Hello and welcome to another episode of Momento Malum today. Turkey has a high level of crop genetic diversity. This is especially true for crops like hazelnuts, sweet and sour cherries, apples, chestnuts, walnuts, apricots, pears, quince, almond, wheat, flax, barley, melon, and cabbage, as well as pulses of many sorts. One of the most important crops in Turkey is the sweet cherry, which grows both wild and cultivated in that region. This species likely has a genetic origin in the regions near the Caspian Sea and Black Sea. The wild sweet cherry populations are potentially useful as a means of adding genetic variation. Therefore, knowledge of the genetic variation within wild sweet cherry could be used to improve the crop's flavor, resistance to diseases and pests, and increase the overall durability of the domesticated cherry. To determine the genetic diversity, several universities from Croatia and Turkey would analyze the genetic variation of 18 wild sweet cherry trees from a wide array of environments using the SSR genetic marker technology. And here are the results. A wide array of variation was found in the populations of wild cherry. The populations grouped into 7 clusters. Group 1 consisted of the tree population, labeled YK11. Group 2 contained the population YK18. Group 3 contained YK01 and YK06. Group 4 was made up of YK09 and YK14. Group 5 contained the majority of the tree genotypes. These were YK02, YK05, YK13, YK15, YK17, YK07, YK08, YK10, and YK16. Group 6 consisted of the populations YK04 and YK12, and Group 7 consisted of a single group, YK03. The researcher found that overall, there was a high level of genetic variation. The genetic variation within the population was recognized as 61% genetically variable. The cultivars also clustered into 7 groups that can be seen on screen right now. Group 1 was the most genetically diverse of these populations. The groups differed from each other, but the groups 2 and 7 had the greatest genetic distance from one another. On the other end of the scale, groups 1 and 3 differed very little genetically speaking. The populations grouped largely based on geography, but the genetic analysis also shows levels of genetic exchange between populations, likely caused by migration between regions and tree exchange. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.